Hey guys, for problem 2 from the last video, I forgot to draw in the cysteine bonds between the two peptide strands, so you're going to want to make sure you do that on the exam. I just forgot. I'm going to be right back and draw all these boxes. Alright, so our protein is found to contain several cysteine bonds. So right off the bat, you can write out your 3 cysteines, just so you can remember. And then it also says that treatment of the original peptide with Cleland's reagent yields a nana, a hepta, and a hexapeptide. And I already drew out the boxes for the nana, the hepta, and then the hexa. And then treatment of the protein with danzyl chloride yields a proline, a histidine, and an aspartate. And danzyl chloride is an N-terminal identifier, so in one of these 3 N-terminals, we're going to have a proline, histidine, or aspartate. And then the next clue says that treatment of the original protein with hydrazine, which is a C-terminal identifier, yields glycine, tryptophan, and asparagine. So we know that these C-terminals are going to have one of those amino acids. Alright, so further treatments of the hexapeptide. Treatment with asp-N protease yields 2 tripeptides. And what asp-N protease does, it cleaves glutamate and aspartate at the N-terminal. So the only way to get this into 2 equals of 3 is if it cleaves the N-terminal of this amino acid. That's the only way you're going to get 2 equal halves. So this is going to be either a glutamate or an aspartate. You don't know which one yet. And it says that treatment of 1 tripeptide with cyanogen bromide yields a mono and a dipeptide. So we don't know which tripeptide that the methionine is in. So there's a bunch of potential spots that we could have this methionine. It could either be here, which it's not going to be because we can only have a proline or a histidine or an aspartate in this spot. It could be here, which is possible, to cleave this into the dipeptide and this into the monopeptide. It could be here, but it's not going to be because this has to be a glutamate or an aspartate. Or it could be here. So since we know it can't be either here or here, it can be in either one of these 2 spots, but we don't know exactly which one yet. So let's read the next clue. It says hydrolysis of the other tripeptide yields a glycine, a glutamate, and a valine. So we know that this is either a glutamate or an aspartate, and this clue tells us that we have a glutamate in this amino acid, or in this peptide. So we know the glutamate goes here, and we don't have an aspartate. And this must be the other tripeptide. So methionine can't go here, and methionine must go here. Alright, it also says that this tripeptide has a glycine and a valine. Now the valine can't go on this C-terminal because we know that the C-terminal must contain a glycine, a tryptophan, or an asparagine. So this must be the location of the valine. And that only leaves glycine to go here. Now we can cross out glycine because we know that this is going to be their tryptophan or asparagine on these 2 peptides. Alright, so treatment with S. aureus V8 yields a mono, a di, and a tripeptide. So S. aureus V8 cleaves at the C-terminal of aspartate and glutamate. So I'm just going to clean this up a bit. Alright, so the S-aureus V8 is going to cleave on the C-terminal of this glutamate, which is going to make our dipeptide. And to get the mono and the tripeptide, we can either have a glutamate or an aspartate go here, or it could go here. Because if it was here, this would be the mono, this would be the tri, this would be the di. And if it cleaved here, this would be the tri, this would be the mono, this would be the di. But you have to remember that there's a cysteine in each one of these three. And we know the cysteine is not in the first amino acid because that has to be a proline, a histidine, or an aspartate. So we know that the cysteine has to go in this spot. So that cancels out the aspartate of the glutamate, which leaves only an aspartate or a glutamate to go here to cleave this into the mono, the tri, and the dipeptide. And we know that this can only be an aspartate, a histidine, or a proline. So this must be an aspartate, and it's not a glutamine, or glutamate, excuse me. So we have the first one figured out, and we can cross out the aspartate up here as well. So for further treatments of the heptapeptide, it says that treatment with endoproteinase lysine C and cyanogen bromide have the same effect, a di and a pentapeptide. Endoproteinase lysine C cleaves lysines at the C-terminal, and cyanogen bromide cleaves methionine at the C-terminal. So in this heptapeptide, to get it split into a di and a pentapeptide, we can either have the methionine and the lysine be here, and this would be the dipeptide, this would be the pentapeptide. We could also get it to go here. This would be the dipeptide, and this would be the pentapeptide. And it says that treatment of the heptapeptide with pepsin yields a mono, a di, and a tetrapeptide. Now pepsin cleaves at leucine, phenylalanine, tyrosine, and tryptophan at the N-terminal. So to get a monopeptide using pepsin, we would have to cut either here at this N-terminal to get the monopeptide, or we would have to cut here. And this spot is already reserved for either a methionine or a lysine. So we have to cut here with the pepsin. And pepsin cuts leucine, phenylalanine, tyrosine, and tryptophan. And we have a tryptophan that we need to put in one of these spots. So the tryptophan must go here, because that's the only spot where we can get the cleavage of a monopeptide. So we can cross out tryptophan, and that means the asparagine must go here. Now to get a di and a tetrapeptide out of this, using one of those 4 amino acids, cleaving at the N-terminal, we could either have one of those 4 go here, which would make this be the dipeptide and this would be the tetrapeptide, or we could also have it cleave here, which make this be the di, this would be the tetra, and this would be the monopeptide. And we know that this spot is reserved for a methionine or a lysine, so we know that one of those 4, leucine, phenylalanine, tyrosine, or tryptophan, has to go in this spot. So I'm going to draw those out off screen. And it also says that hydrolysis of the tetrapeptide yields a cysteine, a leucine, and a methionine. So we know that rules out all these 3 amino acids, and it leaves the leucine. So this is a leucine. We also know that this is the tetrapeptide because that was the only spot it could be. And it says that this tetrapeptide contains the methionine and the cysteine. So that means methionine must go here, and then the lysine must go here. And we also know that we have a second cysteine in this. We don't know where it goes yet, so I'm just going to write that a cysteine could go here, or it could go here. Alright, now that we've gotten as far as we can with this heptapeptide, we're going to move on to this nonapeptide. It says treatment of the nonapeptide with pepsin yields a hexa and a tripeptide. And pepsin cleaves at one of the 4 amino acids, leucine, phenylalanine, tyrosine, and tryptophan, at the N terminal. So to get a hexa and a tripeptide out of this, by cleaving at the N terminal, we can either have one of those 4 amino acids go here, which would make this be the tripeptide, this would be the hexapeptide. Or it could also cleave here, making this the tripeptide and this the hexapeptide. Now the next clue says that treatment of the hexapeptide with PITC yields proline. So the 2 potential hexapeptides that we have are here or here. So PITC being an N-terminal identifier means that proline either goes in this location or it goes in this location depending on where the pepsin cuts at. Now, if the proline cannot go here because pepsin only cuts at the leucine, phenylalanine, tyrosine, or tryptophan, so we know that to have an N-terminal of a proline, that it has to go here. From there, we can determine that this is the histidine, and we can also determine that the pepsin is cleaving at this amino acid. And I will be right back while I write out the 4 potential amino acids that could go here. Alright, so the next clue says that treatments of the nonapeptide with cyanogen bromide, which is a C-terminal cleaver at the methionines, yields a di, a tri, and a tetrapeptide. Now, looking at this, there are many spots where it could cut it into those various sizes, so we're going to move on to the next clue to see if we can narrow it down. The next clue says that hydrolysis of the tripeptide yields asparagine, lysine, and tyrosine. Now, we already know that the asparagine is in this 3rd spot, so this must be the location of one of the methionine cleaves right here, because the tripeptide that methionine cleaves contains the asparagine. And we also know that this tripeptide contains a lysine and a tyrosine, so that narrows down which one of these 4 amino acids goes here, which is the tyrosine. And then this also contains a lysine, which would go here. Alright, so now that we have this methionine cleaving the tripeptide, it also cleaves a di and a tetrapeptide. So to get this into a di or a tetrapeptide, the methionine could cleave here, making this the dipeptide and this being the tetrapeptide, or it could cleave here, where this would be the dipeptide and this would be the tetrapeptide. And then it says that treatment of the tetrapeptide with subprotease yields 2 dipeptides. Now, subprotease cleaves that arginine at the C terminal. So to get either this pentapeptide, because there are 2 potential pentapeptides, this is the first one, and then this is the second one. So the subprotease cleaves at the C terminal. So that means, where's my cursor? The arginine would have to go here to cleave this pentapeptide into 2, or it'd have to go here to cleave this pentapeptide into 2. It says treatment, the next clue says treatment of the nonapeptide with trypsin yields a mono, a di, and a hexapeptide. Now we know that trypsin cuts at the C terminal of lysine and arginine, and we have a lysine in here, so the trypsin is going to cut here, which would make our monopeptide, but it also makes a dipeptide and a hexapeptide. And we know that trypsin also cleaves the arginine, and we have an arginine in this spot or in this spot. Now if the arginine cleaved here, we would get a dipeptide. We would get a a hexapeptide right here, and we would get our monopeptide. But if the arginine cleaved here, we would only get a tetrapeptide, a tripeptide, or another tetrapeptide, and a monopeptide, which is not what we want. So we know that the arginine must go here, and the methionine must go here. Okay, so this next clue says that treatment of the hexapeptide with PITC yields proline. Now the hexapeptide is here, and PICC is an N-terminal identifier, so that means this N-terminal of that hexapeptide is proline. And then we only have one spot left, and we know there's a cysteine in this, so the cysteine has to go here. And for the last clue, it says the nonapeptide forms a disulfide bond with the first cysteine in sequence on the heptapeptide. So from this clue, we're left to assume that there's multiple cysteines on the hexapeptide, and there's only 2 spots left. So we can fill in a cysteine here, and we can fill in a cysteine here. Alright, now I'm going to clean this up and draw in the disulfide bonds. So since the nonapeptide forms the disulfide bond with the first cysteine in sequence on the heptapeptide, you would draw it like this. And that leaves this cysteine to bond with this second cysteine. Alright, if you guys have any questions, feel free to email me. I know these problems are kind of confusing, so if anything's not clear in the video, just let me know. Thanks.